Hi, today we're going to talk about DNA replication. First we want to review a few things about DNA. We notice that this is the backbone. It's made of a phosphate group, pentose sugar for DNA, it's deoxyribose, and we have 4 base pairs, nitrogenous base pairs. A pairs with T and C pairs with G. And notice that there's 2 hydrogen bonds between A and T and 3 hydrogen bonds between C and G. Notice the DNA helix is wound. And then the way the carbons are is very important on the sugar. We label them 1, 2, 3, 4, and 5. The 5th carbon tells you it's the 5' end, and then the 3rd carbon down here will tell you it's a 3' end. Okay, and one thing that's important is 5 to 3, 3 to 5, they are parallel, but they're anti-parallel. Okay. The first enzyme that's going to come into play for DNA replication is helicase. He's going to come in and break these hydrogen bonds, separating the DNA parent strand. Here, helicase is shown. The next thing that's going to happen for DNA replication is the SSBP, single-stranded binding proteins, are going to attach. Because this DNA wants to snap back together, these are going to allow it to hold it open. This opening will create a circle, and that will be the origin of replication. Okay? And the next thing, once this is held open, primase, the next enzyme, comes in and lays down an RNA primer. Okay? For RNA primer, these are RNA bases, which are A pairs with U and C pairs with G there for those. Okay. With this, it's important to know what's happening. So we have this whole circle here. And then we have different ends. If we have this is the 5' end, and this is the 3' end, we know that this one would have to be 5, and then this one would have to be 3. Okay, they're anti-parallel. Okay. So again, this is the origin of replication. Let's move in. Okay. First thing that happens is primase lays down the primer, okay, on the template strand. Okay. Primase is the enzyme that lays down the primer. Remember, the primer is RNA. Okay. Once RNA primer is laid down, DNA polymerase III comes in and he finds the primer and starts to build. Now the building direction is very important. He can only build 5' to 3', okay, which makes this, again, anti-parallel.
Oh, hey, dude. I just came to get my...
Okay, then we got these Okazaki fragments that will then form. So we got 5' here and then 3' there.
Make Kitty proud.
Okay, and it's going to come back and then do the same thing here, and then we got these fragments. Once the fragments are here, we have DNA polymerase I, which will come in and remove the RNA primers. Okay, RNA primers get removed. Then what you're going to have is, at these fragments, there's going to be a little gap that forms. That gap that's formed is taken care of by ligase. Ligase will actually covalently bond those nucleotide bases together. Okay. So, again, just to recap what's going on, we have helicase coming in, breaking the hydrogen bonds, opening up the DNA. We have the single-stranded binding proteins keeping the DNA open. We have primase coming in, laying down the primer, RNA primer. After that, DNA polymerase finds the primer and starts to lay down the nucleotides, A, T, C's, and G's, the ones with corresponding base pairs. Then DNA polymerase I comes in and removes the RNA primer and puts in the appropriate DNA base pair. After that, the gaps where it was fixed are then annealed covalently with DNA ligase. Okay? And then the important thing here is there's a leading and a lagging strand. And what does that mean? Well, since we know that DNA can only go from 5' to 3', there's a certain direction that can be built. And if we know this top strand is 5' to 3', then this is going to be 5 to 3. Okay? And it can easily be built continuously. That's the leading strand. The lagging strand is a little harder to do. So we have 3' here, and then 5' here. So as this is opening up, we can only build 5 to 3, 5 to 3, 5 to 3. And this will be the first section that's built, the second, the third, the fourth, the fifth, and it just has to keep coming back and rebuilding. And it lags behind the leading strand, therefore it's called the lagging strand.